The goal of NeuroDOT processing is to create a topographic map of brain activation, starting with optical array data and ending with a volumetric reconstruction that is ready for visualization and statistical analysis. We can organize the steps included in this pipeline into three broad categories: pre-processing, which refers to preparation of the array data; reconstruction, which requires the creation of a light model applied to the pre-processed data; and post-processing, describing steps following light modeling which generate the final result. In this tutorial, we present these operations in detail. We will follow the flowchart shown here, which appears in the PowerPoint documentation included with NeuroDOT and lists the individual operations comprising the workflow. The presentation is not focused on any one implementation, but rather describes processing steps common to MATLAB, Python, and cloud-based processing. However, the PowerPoint tutorials follow the code more closely and are intended to be used with a specific implementation. Before presenting details of the individual processing steps, it's helpful to consider an overview of the processing workflow. We begin with data provided by an optoid array, organized as a collection of emitter detector measurements of light intensities recorded over the duration of your experiment. The vertical rectangle drawn here represents all data provided by the array. The first step is to identify and exclude unusable measurements. This is accomplished by applying established data quality metrics and removing measurement pairs that fail this evaluation. Data that passes QA nonetheless contains a mixture of signal and noise, and the latter must be removed before the data can be used in reconstruction. Because multiple types of noise are typically present, multiple denoising strategies are employed, such as filtering, regression, and frame censoring. Next, the denoised data is combined with a light model to generate a volumetric reconstruction of light absorption. Lastly, the absorption map is converted into a map of brain activation, which is typically normalized to MNI space to permit group analysis. As introduced earlier, we group operations as pre-processing, reconstruction, and post-processing. We now describe these stages in depth, proceeding sequentially through the processing flowchart, offering background, motivation of the steps, and qualitative explanations of algorithm details. An issue we must confront is the large number of parameters involved in processing. This includes simple settings such as filter cutoffs and thresholds, but also the order of the operations and even the operations themselves. Recommended parameter values are described in the PowerPoint exercises. Here, we only note the default settings reflect best practices that have been found to work well with typical high-density array data. As always, you may contact the development team with any questions regarding the use of NeuroDOT for your specific application. Pre-processing begins with a log-mean transformation of the data. Each source detector pair is divided by the mean of the measurement across all frames, and the logarithm of the ratio is used in subsequent steps. This conversion is required because of the write-off approximation used in light modeling. See the light modeling tutorial for a full discussion. Next, data that should be excluded from the reconstruction are identified using the results of a data quality assessment. Classification is based primarily on two metrics: a maximum usable source detector separation distance and the measurement noise, quantified as its standard deviation. However, the full QA evaluation uses a combination of time domain, frequency domain, and statistical analysis. Additionally, NeuroDOT generates a series of summary figures, such as the example shown here, that allows tuning of the QA parameters to achieve optimal results. The end result is a keeper list, literally, a vector of source detector pair indices. Only these measurements are considered in subsequent processing. Denoising of data on the keeper list begins with bandpass filtering. Previous investigation has established that optical signals reflecting neural activity occupy a frequency band centered at approximately 1/10 Hz. Lower frequency content reflects 1/f noise characteristic of biological systems, and higher frequencies are contaminated by the cardiac pulse and other non-neural signals. These noise sources are removed using a combination of high-pass and low-pass filtering. This proceeds in two stages. Preliminary filtering removes the low-frequency content, but preserves the cardiac pulse. This prepares the data for the next processing step of superficial signal regression. Light used to interrogate the cortex must cross the scalp and skull as it enters the head, and likewise must cross the skull and scalp prior to arriving at a detector. As a consequence, the neural signals of interest are contaminated by vascular signals in the scalp and skull. These generally have similar spectral content, and so cannot be separated by simple filtering. Instead, a regression technique is used. The vascular signal can be estimated using short-range measurements because light traveling from a source to a proximal detector remains close to the surface, and so carries little if any cortical signal. A regressor representing the superficial signal is computed as the average of all nearest neighbor pairs in the array. This regressor is then correlated with all measurement pairs. Finally, the weighted superficial signal is subtracted from each measurement, and this residual is used in subsequent processing. The results of superficial signal regression can be quite striking. The example shown here is taken from a 2010 study that examined responses to a rotating visual grating. As can be seen, there is a notable improvement in localization following SSR, in which lateralization of the response obscured in the unprocessed data becomes clearly evident. Analogous improvements were realized in the CNR of the reconstruction as well as the SNR of the time series response. Following SSR, a second low-pass filter is applied to remove any non-neural physiological signal or other artifacts that may remain. The data now represents the highest quality array measurement pairs, optimized using established spectral and spatial properties of any neural content, so that the subsequent reconstruction will produce the best possible tomographic map of cortical activity. The final step in preprocessing is to downsample the data. Best practice in data acquisition is to sample at the maximum rate that can be sustained by the hardware. For contemporary HDDOT systems, sampling rates of 10 Hz or more can typically be achieved. However, the hemodynamic coupling which underlies functional optical imaging is relatively sluggish. This is demonstrated in the spectral content of the data, which shows neural activity is centered in a bandwidth of about 1/10 Hz. There is little reason to retain oversampled data given the longer processing time and larger file sizes that would result. As such, it is standard practice in NeuroDOT to downsample the data to 1 Hz at the end of preprocessing. An example of data improvements typical of preprocessing are shown here. These results are summarized using a carpet or power plot developed for functional MRI, in which time series data are converted to a grayscale value and displayed as an image. The rows are organized by increasing source detector separation distance. The plots at upper left and lower right show the data before and after preprocessing. 3 effects are notable. First, all data generally appear smoother following preprocessing, with isolated artifacts being removed or reduced. Second, the task response is more readily apparent, especially in the medium distance measurements. This is consistent with the imaging physics described earlier, which argued the cortical signal is carried primarily by medium-distance measurements because light traveling to a proximal detector remains close to the surface, while long-range measurements suffer a substantial decrease in signal-to-noise. Third, not all artifacts have been eliminated. A close inspection of the gray plot reveals several frames contain bright stripes spanning multiple channels. As these do not coincide with the task response and extend into the short-distance measurements, it is unlikely they reflect neural activity. Rather, they more likely reflect some extraneous source. These artifacts can be quantified by computing the global temporal variance of the data, a metric analogous to DVARS that is used in functional MRI. Plotting GVTD averaged across all channels summarizes the time course of frame-to-frame changes in light intensity across the optode array, with large values indicating the presence of artifacts. A before and after comparison demonstrates that preprocessing has substantially reduced GVTD in this example, although a few peaks remain. These likely reflect subject motion that occurred during data collection. GVTD can be used for supplemental data denoising. A threshold can be set, and frames violating the threshold deleted. Alternatively, the measure can be included as a nuisance regressor in subsequent statistical analysis to reduce the effect of these artifacts when fitting a model to the data. Once preprocessing is complete, the data is used to generate the first of 3 volumetric reconstructions that appear in the pipeline. This operation converts light measurement pairs into a map of time-varying optical properties defined on a finite element mesh modeling the brain and surrounding tissues. We assume the optical property of interest is a change in light absorption, indicated by mu sub A. This implies the data were collected using a continuous wave system. Alternative techniques such as frequency or time domain sampling estimate scattering in addition to absorption. These methodologies will be covered in separate tutorials. Parenthetically, the mesh shown here is only meant to be suggestive, as an actual mesh used in light modeling does not include deep brain structures, and the high resolution required for acceptable accuracy results in elements that would be difficult to see when drawn at this scale. A finite element mesh used in the light modeling PowerPoint tutorial is shown in this slide for comparison. Mathematically, reconstruction is nothing more than a matrix vector multiplication. The matrix appearing in this expression, referred to as a sensitivity matrix in the NeuroDOT documentation, is generated using a computational model of how light propagates inside the skull. This is a critical component of the processing workflow. However, construction of the light model is non-trivial. In order to keep this tutorial to a manageable length, light modeling is covered in a separate video. This allows us to move on to post-processing of the light modeling results. The goal of post-processing is to convert the volumetric map of absorption generated by reconstruction into a map of bold activation, ready for visualization and statistical analysis. For reasons that will become clear, the first step is referred to as spectroscopy in the NeuroDOT documentation. To explain this, we must briefly return to reconstruction. As described, reconstruction converts paired light measurements into a volumetric representation of light absorption. However, this is performed not once, but at least twice. Data is collected using at least two light wavelengths. For illustrative purposes, we will assume wavelengths of 750 and 850 nanometers. But the details are, of course, system-dependent. Reconstruction thus provides two volumetric maps of absorption, one at each wavelength. The rationale for carrying two wavelengths throughout acquisition and processing is due to the effect of hemoglobin on light absorption. As can be seen from a plot of the extinction coefficients, light absorption is affected by both oxy and deoxyhemoglobin, so a change in absorption measured at a single wavelength cannot uniquely determine the concentration change in each species. Put simply, we have two unknowns, so we require two equations. The problem is most accurately solved by choosing wavelengths that straddle the isobestic point. The molar extinction coefficients at these wavelengths are available in standard resources, and the relation of interest obtained by inverting a 2x2 matrix. Thus, spectroscopy converts two volumetric maps of light absorption into volumetric maps of concentrations of oxy and deoxyhemoglobin. The final post-processing operation spatially normalizes the volumetric map of hemoglobin concentrations to a standard coordinate space. This is usually done so that individual subject data can be combined in a group analysis. The coordinate system used in NeuroDOT is based on the MNI-152 atlas. Normalization is performed using an affine transformation applied to the volumetric reconstruction, so that the origin, orientation, and bounding box of the geometry matches the atlas. Note, normalization does not include a nonlinear or warping transform as typically done in functional MRI. This is because structural information is not provided by optical imaging. To summarize, post-processing involves two data transformations. The first converting a map of absorption changes into concomitant changes in hemoglobin concentrations, and the second normalizing the map so that it uses an atlas coordinate space. The reconstruction is based on paired light level measurements provided by the optoid array, which have been prepared using a standard series of pre-processing operations. This following a quality assessment which identifies and excludes unusable data. And so we have come full circle, describing the complete NeuroDOT processing workflow. The end result is a 4D tomographic map that is ready for statistical analysis, conversion to a standard file format for export, or review using either NeuroDOT or external visualization tools. This video will have prepared you to work through any of the PowerPoint data processing tutorials shown here. These provide self-guided exercises, applying the workflow described in this video to generate a tomographic map using sample data and a predefined light model. Additional information may be found in the resources listed here. Feel free to contact the development team with questions or post them in the comment section below. Thank you for your interest in NeuroDOT.